begin.
I'm Amy. I'm Stephanie. I'm Janet.
So,
And this is our project, and we called it the dessert because of the materials used.
And it's expressing protein synthesis.
Okay, so protein synthesis basically has two parts, and the first part, what we're going to talk about, is called transcription. So transcription happens in the nucleus of a cell, and it's basically the catalyzation of RNA from DNA, the template. And the enzyme that helps with this catalyzation is called the RNA polymerase, which is represented here. And the way it helps is that it first goes to a promoter, which is a sequence of nucleotides that tell the RNA polymerase to bind here and start here, basically. And so as the RNA polymerase is going down, the DNA is beginning to unwind.
And then from there, the RNA polymerase is moving down and it's adding free-floating nucleotides, and that will form a new RNA strand. So these hydrogen bonds, which are represented here, are going to, these are unwinding. These are unwinding. This and a new RNA strand is going to come in over here. The difference here between the RNA strand and the DNA strand is in the DNA strand, you have a thymine is complementary to the adenine, but in the RNA strands, uracil is complementary to the adenine. So for example of this, we have ATC. So for example, in the RNA, it would be UAG.
Okay, and another difference from this to DNA replication is that transcription only occurs in one specific section on one DNA strand, and so that's a difference. And as the RNA polymerase is going down the line and past what it's already added for the RNA strand, the DNA begins to rewind again.
So imagine it more like that, so it's binding again. And then we have, eventually the RNA polymerase is going to reach the termination signal, which is a part of the sequence of the DNA, which tells the RNA polymerase to stop creating complementary to nucleotides that are complementary to the DNA.
And so when the RNA polymerase reaches the termination signal, it basically comes off and becomes free-floating and can attach to another gene. And then the RNA is made and released, and so the DNA is back to its original shape of being, with the hydrogen bonds and everything, it's back to its original shape. And the RNA here can be used for the next part of protein synthesis, and this can either be mRNA, which stands for messenger RNA, tRNA, which stands for transfer RNA, or rRNA, which stands for ribosomal RNA.
Okay, so the next step of protein synthesis is translation. And so that's when this RNA strand, it comes out of the nucleus, and it's in the cytoplasm right now. And so in the cytoplasm, these tRNA strands, or these tRNA, shown by the T-shaped cookie, these tRNA have the complementary nucleic bases to the RNA, and attached to them is an amino acid shown by fruit in our case. And so these tRNA with the complementary RNA strand, they attach to this RNA, to what their complementary RNA is. And then as they connect, the tRNA leaves, and a new tRNA with another 3 amino acids comes in. But while this is happening, this has to be done by, it can't just do it by itself. It has to have something that does it for you. So this is going to be shown by the ribosomes. So the ribosomes, they aren't actually one part, they're actually 2 parts, 2 subunits. And so the ribosome, it will be able to bind the amino acids together. And so as these amino acids bind together, a protein is formed.
and so once all the amino acids bind together by polypeptide bonds, they compact and fold together into any shaped proteins, which we used as fruits. And I also like to mention that the process of translation does not start unless there's an AUG codon, and so yeah, the translation does not start unless there's an AUG codon.
And then of course, it stops with the stop codon, which is shown by UGA in our case. So that is our dessert of.
So that's our representation of protein synthesis.